inauguration are Dr. Sanjay More and Dr. Anil Gaikwad. I request our chairpersons to please come on the dais. Sanjay More is the Dean at Government Medical College, Parbhani. He is the alumni of this own BJ Medical Institute, and he has worked in various institutes like in Aurangabad and all over the Maharashtra. Dr. Anil Gaikwad is currently working as Associate Professor at Department of Microbiology, Government Medical College, Aurangabad. He is the member of MUHS Task Force for Microbiology CBME curriculum. With 22 years of teaching experience and 17 national and international publications to his credit, he continues to guide us as expert for different state level committees. Sir, Dr. Anu Panvikar.
Namaste and good morning to all. Dr. Anu Anvikar sir is an MBBS MD microbiology doctor from GMC Aurangabad. He had done excellent work in the microbiology field, and he was director previously for the National Malaria NIMR, that is National Institute of Malaria Research, and he had developed one center in the Southeast Asia, which is only third by the WHO. And also, sir had done excellent work in the tribal health in Jabalpur area. Also, sir is right now director for National Biological Center, and sir has the excellent achievement. Sir has had 7 patents on his name. That is the extraordinary work done by the sir. Sir got 2 prestigious national awards for his work. Now I request all to clap for the sir, Dr. Anup Anvikar sir. Please sir, welcome and deliver your lecture to us. Thank you sir.
Thank you, my chairs. I feel proud to be among my respected teachers, my colleagues, students. So it reminds me of one conference which we had hosted in 1997. So, and most of these stalwarts were present at that time also. Bharadwaj madam, Narang madam, Bansal madam, Karyakarte sir, of course, Jagirdar sir. So it really feels proud to be among my teachers again. I am thankful to the organizing team for inviting me here, and especially Karyakarte sir. I took little time in deciding the topic. Considering the theme, which was molecular epidemiology, I thought whether I should work on malaria, in which I had worked for almost 20 years, or should I talk on COVID. But then, being the ambassador of the biologicals, I thought I must talk on biologicals. Initially, I thought it is a microbiology conference, but then there is a lot of microbiology, immunology in biologicals. And I especially wanted the youngsters to know about the biologicals, biotherapeutics, and maybe few of them can join the field of biologicals in whatever capacity in future. So I will talk about biologicals. After a brief introduction, I will talk about why it is extremely important to monitor the quality of the biologicals. Then about my institutes and what are the challenges in their regulation and how we can move ahead about that. So the biologicals, or they are also called as biologics, and most of the biologics, they also fall under the category of biotherapeutics. So they are a relatively newer category of the therapeutic agents. They target specific markers, unlike the chemical entities, and they are the future of medicine due to their targeted efficacy and extreme safety. Say for example, if we compare this with a chemical entity, so in cancer, we are giving chemotherapy. The drug, it is a chemical. It does not recognize the cancer cells as a separate entity. And in turn, it also damages the host cells, which are normal also. But unlike that, biologicals will specifically target the cancer cells. The figure on the right hand side, it shows the example of a monoclonal antibody which is targeted against the cancer. And the monoclonal antibody, it acts in all different ways. So it will either cause the antibody dependent or the complement mediated cytotoxicity, or it will act through the different immune effector cells. We can conjugate the monoclonal antibodies with the specific agents, maybe some chemical or some specific agent which will act just on the target cells. Humans have been using the biologicals since millennia, and we have been manufacturing them for decades, but we have only recently come to know about their true potential, but then there is still a long way to go. And the basic difference again between the chemical and biological entities, they are derived from the natural sources, unlike the chemical entities. And those sources could be human, animal, or from the different cells or the microorganisms. And they contain essentially sugars, proteins, nucleic acids, cells, etc. And their production is challenging as compared to the chemical entities. And we need to use advanced biotechnological methods or other cutting edge technologies in order to manufacture them. So these are large, complex, and often consist of heterogeneous mixtures. And they are generally made in genetically engineered cells, and the cells itself, they themselves have a lot of variabilities. So our biologicals are also going to show variability. That's why it is very important to take special care of the biologicals. And even if there is a very minor change in the manufacturing process, that can cause significant changes in the efficacy, immunogenicity, and also safety. We will talk about that a couple of minutes later. So if we compare aspirin, so the molecular size is around 180 Daltons or 0.18 kilodalton. And if we talk about a typical monoclonal antibody, the size is 150 kilodalton. Aspirin contains 21 atoms, and a typical monoclonal antibody contains over 20,000 atoms. It's like comparing a bicycle with an aeroplane. So we are talking about the aeroplanes. So again, to show you the difference between the chemical and the biological entities, the chemical medicines are synthesized, while as biologics are grown. So if we again take the example of aspirin synthesis, we are just making a reaction between salicylic acid and acetic anhydride. And what we get is salicylic acetyl salicylic acid or aspirin. So it is as simple as this. But when we compare it with a typical biological, say let us take example of a vector vaccine. Earlier we talked about the monoclonal antibody. The processes, they essentially remain the same. So here we have got this chimpanzee adenovirus which we are modifying, and then we are identifying some required genetic structure, and then we are inserting it into the vector. And then this is also not done. This will, when injected into the body, it will elicit the required immune response. So the process is such a complex one. These are the examples of some of the biologics: vaccines, blood components, blood products, monoclonal antibodies, which are used in the treatment of cancers. Basically, we are quite advanced in that particular therapy. Of course, there are monoclonal antibodies targeted against other diseases too. Then allergens, somatic cells, cell therapy, gene therapy, tissues, recombinant therapeutic proteins, diagnostics, and n number of other biologics. So there could be the coagulation factors or the blood products and n number of biologicals. And which are the sources for these biologicals? They could be made up from cell culture or transgenics or enzymes, hormones, this insulin, streptokinase, these are also biologicals. They could be recombinant proteins, cytokines, monoclonal antibodies, we have seen the example, allergenic extracts, cell therapy, gene therapy, xenotransplantation products, vaccines, recombinant DNA products, stem cells, cell-based biologicals, blood products, and the list is continuing. I would like to apprise you about one small terminology. It is called as biosimilar. What is a biosimilar? It is a biologic only, but there is a little difference from the reference product or the innovator product. And why we manufacture biosimilar? What is the need? Because whenever there is a biological which is an innovator product, there is a lot of cost required in the development of this biological, both in the laboratory as well as the clinical development, and millions of dollars are required for that. We may compare it with the generics. So biosimilars, the process of approval is not that stringent. You just need to demonstrate equal safety and efficacy. And we have tweaked the original or the innovator biologic a bit. It will be little different from the biologic, but it will have a similar action. Let us take the example of the anti-CD20 rituximab biosimilars. So we have compared 3 different biosimilars of this innovator molecule by different methods. So we have done this in our lab. So just one example, by HPLC, we can see a heterogeneity in case of the charge variants of this rituximab biosimilars. And there are variations in the acidicity as well as the basicity. So when we analyze them by HPLC, we will see the variations. But when we talk about their biologic activity, say this is one example of the complement-dependent cytotoxicity assay. So these all 4 are falling in the same line. So they have got similar biologic activity. So they fall under the category of biologicals. So they are little affordable compared to the biologicals, and they also have a similar efficacy. But then yes, we need to monitor their quality from time to time. There is one more terminology. It is called as bio-better. So these bio-betters are the next-gen biologicals. They have the same target or the mechanism of action like the innovator molecules, but since there will be some structural changes, and these are the improved formulations, and they will definitely be giving us increased or improved efficacy. They are not cheaper than the biosimilars because the cost for clinical development again is much more than even the innovator molecule. So why we need, I will sum up again, why we need quality control of the biologicals. So these are large, complex and heterogeneous mixtures. Then their process of manufacture is not like the typical chemical entities. We need to make them in genetically engineered cells, which themselves are variable, and hence we need to assure the quality of these biologicals. Their manufacturing is challenging. Even if there is a little small change, it will lead to some serious consequences. And their production, since it is also an unstable process, they also require special handling and storage. That's why we need to monitor their quality at different levels. I would like to quote one example of little change in the structure. So we use recombinant erythropoietin formulations. So this was in use since years in Europe and Americas. So there was one formulation change in 1998 in this recombinant human erythropoietin. And what did the manufacturer do? They replaced the human serum albumin, which is normally used as a protein stabilizer, with polysorbate 80. And what happened? Due to this polysorbate 80, there was development of neutralizing antibodies in the patients treated with erythropoiesis stimulating agent. And later on, what we saw, these are the cumulative figures, the dark black parts of the bars, we can see that they are almost equal. There is no change. So these are the cases of aplasia, pure red cell aplasia. So in the US, where the original formulation was being used, there were very little cases of aplasia. But in the Europe, this new formulation was used, and they started getting more and more cases of aplasia. So this is the seriousness of quality checks for the biologicals. I belong to the National Institute of Biologicals, which is an autonomous institute under the Ministry of Health and Family Welfare, Government of India. The institute is ISO IEC 170 accredited for the specified scopes. It's also a WHO collaborating center and also a support cell for WHO PQ. What we do? We are into quality control testing of the various biologicals as well as immunological products. What are they? That we will see. Then we also work with various institutes. We have a strong training program for quality assurance of various biologicals, including the blood and the blood products. So these are the different laboratories of ours. So we have got few diagnostic laboratories like the blood reagent laboratory. So these are labs. They will receive the samples of the biologicals. They will conduct different tests. They will test these biologicals with different parameters and then certify these products as pass or fail. In our words, we say it standard quality and not of standard quality. If a product is not of standard quality, that won't hit the market in India. So there is a blood reagent laboratory, immunodiagnostic kit laboratory, molecular diagnostic lab, then biochemical kit lab, COVID kit testing lab, then we have a blood products lab, recombinant products lab, enzyme and hormone lab, then therapeutic antibody lab. We are also into quality testing of the vaccines as well as anti-sera, and currently we are testing 16 different types of vaccines. And we have some support facilities in the form of sterility lab, animal facility, and the bioassay facility. And we conduct the testing for regulatory purpose, procurement purpose by the government, and for the legal purposes. If some biological product is picked up by a drugs inspector, then it reaches us in a cold chain. And if there are some issues, then that product is discontinued. So we are currently testing around 300 different types of biologicals under the various categories which we just saw. And we receive around 2,000 batches of the different biologicals from all over the country. And as we can see, around 3% of the batches, they are not of standard quality, and they do not hit the market. And all sorts of biologicals, they have failed at some or the other point of time. So I will quickly go through few of the testing procedures followed by us. So if I talk about the diagnostics, we undertake the QC testing for the diagnostics of HIV, HBV, HCV, syphilis, dengue, COVID-19 diagnostic kits. Then we also undertake the QC testing of the glucometers, glucometer strips, blood glucose reagents, then the different blood reagents, anti-A, anti-B. And how we are doing that? We are testing them using the characterized panels or the reference standards. And I would like to inform here that Professor Rajesh Karyakarte is on our advisory board for this diagnostics, and he has been providing us useful inputs for improving the testing. So this is the example of one characterization of the COVID-19 panels. A lot has been talked about COVID-19 sequencing, so I have cut short this aspect. So we are characterizing the panels by every possible way. So if I talk about malaria, when the kits are tested in the laboratory, it's not just that the kit will contain parasite. There will be measured level of the HRP2 antigen, LDH antigen, or the enzyme, aldolase. Then by molecular assays, it will be characterized as a mono-infection. Then the sequencing will also be performed for certain things. In this case of COVID, we ensure that we will subject the kit to testing by using the panels of different types, whether it is Omicron or Delta or whatever. That kit should be able to detect that particular variant. And where the diagnostic kits are failing. So I was also thinking when I first went to NIB. So what am I doing? But then what I saw, the factors which are leading to the not of standard quality kits, each one of them is having a big research question. Why the kits are failing, where we need to work upon. And accordingly, we are advising the manufacturers, no, you work this way, so that they improve on their quality. And we can even conduct research to improve upon that. So usually they are failing for the low sensitivity rather than specificity. And at times the reagents are of poor quality, their affinity or stability could be compromised. Then there could be a contamination of the reagents or the transport medium. And at times we have failed the batches of VTMs which showed us some growth. We also subject these VTMs to sterility testing. Then there could be degradation of the heat labile reagents, there could be transport or the storage issues, or there could be the issues related to the structural integrity of the antibodies. And at times the manufacturers, they may not follow the GMP. This was the example of the diagnostics. But when it comes to the biotherapeutics, we subject each biotherapeutic to a stringent testing, and around 20 parameters are tested, mainly for the potency of that particular vaccine or the biotherapeutic, then sterility, identity. So there are around 20 parameters like that. So these are few examples of the biologics and how they had failed, like human albumin, then immunoglobulins, then anti-T lymphocyte immunoglobulins, coagulation factors, different types of them, fibrin sealing kits, and how they failed in parameters like sodium content or total proteins, anti-A, anti-B, hemagglutinins. Then they passed everything, but they will show us the HIV antibodies. So such blood products cannot be administered to our patients. Some failed in potency, pyrogen. Pyrogen testing is very important. So whenever a biologic arrives in our lab, we will inject it into the animals to see what is the effect of that particular biologic. And pH, solubility, thrombin assay, and there are n number of parameters because there are 300 types of biologicals, and multiplied by 20 there will be 600 different parameters. I have just listed few of them here. Sterility testing. Whatever product hits the market, each batch, we are subjecting it to the sterility testing. Mostly in case of the injectable products, and we are using two primary culture media, the fluid thioglycolate medium for the bacteria and soybean casein digest broth for fungi. And we typically incubate them at 14 days, and we follow the different temperatures for these two different media. And the product must be free at the end of 14 days of incubation, otherwise that product fails. We also conduct research to improve our QC testing. This is one example of an infectivity assay. So we have one bioassay lab. So in case of Covishield vaccine, what we do, so this is the typical test which is described. So we will detect the expression of the backbone protein, and we visualize that by dab staining. And you can see on the left-hand side, it becomes a bit challenging to visualize that and also quantify that. But what we did, we used immunoblotting and immunofluorescence, and where instead of backbone protein, we tried to standardize the study of expression of spike protein. So this is little quick, robust, and it's not that subjective like the earlier method. So again, this is one example how we are trying to improve upon the QC testing, and this is the case with each and every biological. We have a research and training wing. So very recently we started the PhD program, and last month only we received 10 PhD students. So we have a PhD program, and it is open to the serving people as well. If they are sponsored, means if they continue getting salary from their institutes, because we can't pay huge stipends to them. We have just now started our own postdoctoral fellowship program. If somebody is PhD already, he is most welcome to join our institute, and we pay the stipend in line with the ICMR or CSIR fellowships. We have our own fellowship, just now started. The advertisement has been floated recently. We have started a certificate course on QA of the biologicals. Then we have got structured training programs, and many of them for MD students as well. They are on the quality assurance. And we have been contributing to papers, monographs, so monographs are similar to the publications, but then they will give us the SOPs for the QC testing of the biologicals. And there are a lot of career opportunities in the field of biologicals. Means it could be quality testing of biologicals, it could be production of the biologicals, then it is research in the field of biologicals, regulatory aspects of the biologicals, and there is a definite need of the manpower, and microbiologists are the one who are going to lead the show. So what are the challenges? Why we can't regulate the biologicals quality that effectively? Because the biologicals are diverse. My institute is just testing 300 types of biologicals. Maybe there are n number of manufacturers for each one of them, but then there are n number of more biologicals for which we do not have capacity. The biologicals are diverse because the raw material is also diverse. There are very limited number of testing labs, so most of the biologicals, we are the only lab in India at the moment. So many a times they need to be approved for regulatory purpose on the basis of their dossier review or the inspections, and a full testing cannot be conducted. For many biologicals, there is no reference standard available. Then if we talk about diagnostics, there is a declining incidence of the infectious diseases, example HIV. So it becomes difficult to get samples for the panel preparations, and for that we are trying to collaborate with more and more institutes. And one more thing, in chemical entities, the drug inspector goes to the place. If there is some suspected spurious medicine, he will just pick up the sample and send it to the lab. But in this case, it is not as simple as that. He needs to maintain a cold chain. So again, the surveys, they are little difficult. So what is the way forward? We need to establish more and more testing labs. If it is not possible, we can motivate more and more institutes to undertake the QC testing. At least they can start with the diagnostics. And I give an open invitation to all of you to start work in this direction, and we will be happy to have you as our partners. We need to have a mandatory QC testing at the pre-marketing as well as the post-marketing stages. At the moment, this post-marketing stage testing is not there due to the deficient capacity in the country. Then parallel pre-release QC testing can also be done to save the time required for standardization of the QC testing. Then when it comes to particularly say biosimilars, there is need to harmonize the testing methods, because if there is one biological and if there are 10 biosimilars, I can't have 11 protocols for the 11 different types of biosimilars. Then we definitely need research at all the levels, and the medical colleges are the best ones to conduct research. I have seen many setups like medical college, blood bank, then health setup, research institute, and now regulatory institute. So I have been at all the different types of places. But what I have seen in medical colleges, there is a huge sample flow, and more importantly, there is guidance of the senior teachers. So the students are under the umbrella of the teachers. So it becomes a smooth journey to conduct the research. And we need to conduct research to improve the QC testing and replace the manual assays with the objective results. So this was all, and I will be happy to answer questions after the judges, after the chairs say something.
Thank you, Dr. Anup. It's a journey of Dr. Anup since when I passed from my MD from BJ and joined as assistant professor at Aurangabad, he was in the final, I think, final of third year of MD. And since then, I have seen the journey of Dr. Anup. According to his nature, which is very simple and straightforward, he has made such a complex biological topic into a very simple language and simple presentation so that we could understand the thing. And really, he had chosen a different path. He was assistant professor nearly for 5 to 7 years rather, but he has again chosen a different path, and he has shown that you can rise up to that level, right, going from Marathwada region to Delhi and heading the institute, which is a national and international report. So all of us can again lead the path and clap for him so that he has risen to such a level. And also, important thing, he has also invited for research and this thing for MD as well as the in-service candidate. So we can think of joining him, joining the institute and taking the advantage of his position to get more research facilities in our medical colleges, which we run, and progress. Really, this is the future rather. In therapeutic, what we do over here in medical colleges, we teach, but implications or applications in the public, applications, your knowledge applications in public for the benefit of public can be really done by research through such institute, taking the help of such institute, and where people are ready to give, we should be ready to take the thing. Okay. So thank you very much, Dr. Anup, to come over here and give such a nice presentation. Thank you.
Well, thank you very much, respected Dr. Anup Kumar Anvikar. This was really an excellent, highly informative and motivating talk. So regarding the word biological, I think whosoever has joined this hall today might be remembering that somewhere we have read the definition of biomedical waste, where the bottom line says that in testing of biologicals. So what is that biological? I think everybody must have got a fair idea about it. Sir, you have very nicely pointed out what are biologicals. You also highlighted the importance of testing of biologicals. You have also made us aware of not only biologicals, but the difference between the chemicals and the biologicals. Not to that, biosimilars and even newer the biologicals. We are very much glad, sir, to know that under your leadership, you are testing more than 300 biologicals. There are lot many challenges also. And if you could see the setup of your laboratory work, I think it is a combination of the whole microbiology. So it is really creditable, sir. Hats off to you. So we are very much happy. And as Dr. More sir has somewhere mentioned about your journey, I can see a lot of budding microbiologists here, and even young teachers are also there. So from both of us and maybe from the organizers, please take our sincere request that please have few comments about your fantastic journey.
Thank you, Chairman sir. You are now reminding me of my past. So I was a PG student in GMC Aurangabad. One fine afternoon, few of the PG students, we were chit-chatting in the corridor and leaving the UG students unattended. So Karyakarte sir was going somewhere, he saw us, did not say anything, we only felt something and we went inside. But I don't remember everything now because it's almost 25, 27 years back. So sir then called us one by one and gave us certain assignments. So he used to publish one monthly wall magazine called as Window. So he gave us that assignment. So he called me and asked me to get one good review paper and write a summary in 1,000 words. And along with many more columns, write down the whole of the thing on the A0 paper size. So initially, I thinking as a punishment, हे काय दिलं सरांनी आपल्याला? So म्हणजे इच्छा नसतानाच मी ते बनवलं. And then I thought, let us take interest into this. Then from second issue onwards, I got interested into that, and let us put on our sincere efforts. And At that time, I also remember my teachers used to tell all of us, "Baba re, tumhala sarkari naukara nahi आहेत सगळ्यांना." Because at that time, there were only limited number of medical colleges. And the nightmare, it came true. I wasn't called by the MPSC. So I thought, let us focus on some other research, because there was always a threat that someone permanent will come and my job is gone, what I will do tomorrow. So I went to research. So there used to be very few medical personnel in the ICMR setup. So I had to do epidemiology. But I found microbiology in that. And then I shifted my institute and I was required to pharmacology, clinical trials, because there was no MBBS doctor also. Then I think what to do? Let us try to find microbiology in that. Or if not, let us add microbiology to that. There was one quote I had read and one of my colleagues had told me in some other reference. So there was one quote in front of a photographer's shop. "We capture your beauty, else we make you beautiful." So I tried to find microbiology in clinical trials, the safety assessment, efficacy assessments. So what is efficacy assessment? So I can add drug resistance to that, which is all microbiology. Or the safety assessment or efficacy assessment, say for example in case of malaria, if I see some prognostic marker or just simply parasite count. So everything is giving me one research question. Then there was public health, because just like MBBS doctor, I have to go there. So I learned public health, did many operational research projects for the elimination of malaria. And in fact, my model in malaria control, it could eliminate 90% of the malaria in Odisha. So I found microbiology in that also. I tried to add microbiology, see the parasitemia. In asymptomatics, I will see the parasitemia, try to have different types of assays. So it was all the learnings during the PG life that all helped me. And in the meantime, I also started working on quality assurance of the malaria diagnostics, which is currently the only WHO lab globally. So these, along with the clinical trials, the pharmacovigilance, so everything helped me to land up in NIB.
Now, any questions from audience are welcome.
Sir, please.
Mic. Give the mic, Dr. Jain.
Sir, I have one small query.
Sir.
We are accustomed to deal with pharmaceutical agents, and in medical pharmacy shops, there is an expiry date to the products which are sold in the across the counter in the medical shops. We have used biological reagents in microbiology laboratories also. There is a shelf life and there is a storage recommendation. In your experience, how do you determine the life of a biological agent that you are investigating, and how do you decide about the plausible expiry of the product that has been tested by you? And do you certify such type of certification that this product needs to be utilized within say 90 working days or say 45 working days, half-life of some biologicals like that? And if so, what are the parameters? How do you utilize what parameters to define this life of the biological?
Sir, thank you very much for this, sir. This is extremely important issue, and if a biological, whatever type of, or even the chemical entity, if the shelf life, say, if it is supposed to be 1 year, then it should have the whole lot of parameters passing the tests even after 1 year. So actually, this is the prerogative of the regulator based on what are the claims of the manufacturer. But if we talk about our role, means we do reserve some samples which are received by us. We retain equal number of samples in our laboratory at the appropriate storage temperatures in order to test them whenever it is required. Say, I will give the example of the malaria diagnostics with which I was dealing before going to NIB. What we used to do, we used to test all the diagnostics with the same set of panels 3 months before the expiry date. And many times it happened that they were not up to the mark. And many times it also happened that if, say, for example, a malaria diagnostic is giving 4+ band on 0 month, it will be just 1+ band, or it will be just strict lines on, say, month 21, which is 3 months before the expiry date. But yes, in case of biotherapeutics, it is highly possible that all the parameters, they are not good with that particular biological at the end of 3 months, or maybe at the end of the expiry time. It is possible, and it is not a routine request, but at times, if the manufacturer or the regulator asks us to conduct a full testing on that particular biological near the expiry, then we do have the capacity and we do undertake that kind of testing also. And not only near expiry, but on many occasions, the Drugs Controller Office sends us the biologicals picking up randomly from the field, and it sends us that as a legal sample, and we test it, and those results are valid in the court of law as well.
Thank you, sir.
Thank you, sir.
I am Dr. Rajendra Surpam. I sometimes deal with ISO 17025. I just wanted one query, that if the reference standard is not available for some of the biologicals, how we are going to test? Number two, you probably maintain the IQC, but is there any provision for the ICAS for your biological? And the third, our experience is that, first of all, I am very much thankful to you for providing a good, nice, quality kind of COVID testings and kits for the Maharashtra government. But the problem is that when we are taking kits or any biologicals or reagents in a bulk, okay, and all of a sudden we got a letter that this lot is probably contaminated or should not be used. So there is a huge, because that manufacturer don't want to take it back, because the costing for the transportation is very, very high. So in that case, they ask us to discard at our level. So what are the guidelines? Is there any guidelines particularly for the biological to discard? That is what my query is.
So thank you, sir. These are extremely important issues. I should have talked about that during my talk itself. So I will go randomly. So first I would like to reply regarding the quality of the diagnostic kits, including COVID-19 or any biological for that matter. If we talk about the regulation is that whenever a new biologic is introduced in the Indian market by an Indian manufacturer, the 3 consecutive batches of that particular biological, they need to be passed from a NCL, like our laboratory. And after that, they are free to sell whatever they want to. I am talking about the indigenous manufacturers. But for imported products, because we don't have control on their manufacturing facilities, we have to test each and every batch. So now what we should do for this? So it is the job of the state governments and the procuring agencies. And we have been writing from time to time. And many states, they have a rule. So whenever the state government is procuring any biological, they will ensure that each and every batch undergoes quality testing at a laboratory like NIB. So I think that is most important, which we need to take into account. So this was the first thing. Second thing, regarding the discarding of these materials. So we need to follow our standard biomedical waste guidelines, and they need to be considered as the infectious agents, and just like we discard the the red bag reagents, they need to be discarded like that, sir.
Okay, we are somewhat ahead of time. Now, thank you, Dr. Anup, for lucid, nice presentation. Thanks the audience for patient listening. Thank you.
request our chairpersons, Dr. Sanjay More sir and Dr. Gaikwad sir, to felicitate Dr. Anup sir. Thank you, sir. Thank you so much, sir. Now I will request Dr. Ashish to felicitate Dr. Sanjay More sir. Thank you, sir. Now I will request Dr. Rahul to felicitate Dr. Anil Gaikwad sir. Thank you, Dr. Gaikwad sir. Thank you, sir.
At this particular conference will be etched on your memory forever. That is why I hope, that way I'm sure about it. But then we need to keep it with us always. So we have a bit change of program. There's a lot of people here right now. We'll all go outside now and we'll have a group photograph. So I request all of you to come out. Thank you.
Teachers who are there in this event. And before I start talking anything about science or about the system or about the technology, let me first pay the due respect to the microbiology community, to all the clinical microbiologists who have played a role which is as important as the soldiers on the battlefield, saving hundreds and millions of lives during the COVID pandemic. So before I start, I request everybody to give a big round of applause for us for the great job which we have done. And I thank the microbiology fraternity, the technical staff, and the upcoming microbiologist for the role. So when we talk about the multi-top technology, or when we talk about the system as such, so before that, when we talk about the clinical microbiology, so very few incidences we have seen that there have been some great or remarkable innovations. So in the 21st century, when we talk about the clinical microbiology, so one of the biggest breakthrough which has taken place is introduction of the multi-top technology in a routine clinical microbiology usage. And in the world, there are two main players, bioMérieux and Bruker. Apart from that, there are other companies also based at China and other places who are providing the solution. So this session will not be a commercial session talking about the commercial aspects or the commercial benefits or the product specific details, but in general, how multi-top can impact the clinical microbiology setup. And one of the biggest challenge which growth-based microbiology has, that is time. So how can we, help the clinical microbiology lab to surpass the challenge of time and provide an actionable report to the clinician? Because that is what we are here for, to provide a report to the clinician which is actionable And on the basis of that, they can act, they can choose the antibiotics, they can escalate or de-escalate the antibiotics. That is the need of the hour. So before I start, let me give just 1 minute review of from where we are. So we are from bioMérieux. bioMérieux is one of the leading microbiology companies in the world. We have 3 main pillars: the immunodiagnostic solutions, molecular biology, we have the film array, and we have the microbiology, which is the main solutions that we provide. So all of us are aware about our microbiology solutions, BacteriAlert, Vitec, and so on. So coming to the brief of what exactly we are going to talk about today. So we are going to talk about the MALDI-TOF system. So bioMérieux has got or introduced a new MALDI-TOF system last year, which is called as the Vitec MS Prime system. Now this Vitec MS Prime system is developed based on last 55 years of clinical experience which bioMérieux has. And I'm really proud to say that bioMérieux has got world's largest repository of cultures which they have collected over decades in them. And so many, when you go and see the ATCCs, and when you see the source of that ATCC strain, you will find that, the source has been given by bioMérieux. And the new system, that is the Vitec MS Prime, which is there, has been developed based on the past 10 years experience collected from the clinical microbiology fraternity and the engineers, that what are the challenges which they have faced, what are the day-to-day issues which the technicians have faced, and we have tried to address them. So MALDI-TOF technology is called as matrix-assisted laser desorption ionization time of flight. So what exactly we do in the MALDI-TOF technology? We take the slide. This is the target slide which we take. So this is the system. It's a benchtop system, and the only reagent which it requires is called the target slide. And how it functions, I'll explain just in a small second. So there is a solid surface on which we lay the microorganisms, and then it is hit by laser. What the laser will do is the laser will charge the matrix and it will charge the ions. The ions will fly in a tube, vacuum tube, and they will reach the detector. And once it reaches the detector, a profile is created. And this profile then later on is analyzed by using different algorithms. So there are different companies, and different companies use different algorithms. So we use a system which is called as advanced bin matrix for our analysis. So one of the biggest benefits of MALDI-TOF is the ease of use and the scope of identification. So when we talk about the biochemical based identification or morphology based identification, the biggest challenge is the scope. But over here in the MALDI-TOF technology or in the system, we can identify more than 1,000 different species of bacteria. We can identify up to 200 different yeasts. We can identify mycobacteria, nocardia, molds, and also brucella up to the species level. And whatever I am discussing is all the FDA list and not the RUO or the research use list. So how exactly it works? So as far as the usage is concerned, it is extremely, extremely simple. There is a uniform process. So there's the target slide, and on that target slide, what you have to do is just pick one isolated colony and place it on the spot. You have to allow the spot to dry for a couple of seconds. As soon as the spot is dried, you have to add 1 microliter of matrix. We use CHCM matrix, and you have to allow it to dry. So the entire process ideally takes less than 30 to 35 seconds, provided you have the isolated colony, and that's all what is required. Once it is there, once it is ready, you can prepare multiple spots and simply go and load it into the system. Once it is loaded into the system, it takes less than 30 to 45 seconds for identification of a single organism. So for a slide of 48 spots to complete, bioMérieux Vitec MS takes less than 30 minutes for the completion. So if you calculate, and if the spot quality is good, then you can get the identification in less than 1 minute. Average time is between 30 to 45 seconds for the identification results. Coming to the other procedure, that is, how do you load it and how do you process it? That is also extremely simple. Just you have to click on the spot and scan the barcode, and on the basis of that, you can process the test. So the entire process, if you take, is less than 1 to 2 minutes. Now, what is the biggest advantage? The biggest advantage of MALDI-TOF technology is the database. So we can identify mycobacteria. There are more than 100 species if you talk about, and when we talk about the FDA approved database, so in that there are close to 50 species. And the best part is that you have to use a single protocol. There are no different protocols of inactivation and extraction. So it is very important to inactivate the mycobacteria, because we don't know, are we dealing with MDR or XDR strains? So the first thing is complete inactivation and then extraction. So the inactivation extraction takes up to 40-45 minutes. As soon as it is done, you can process it. And bioMérieux is the first company to get a FDA approval for Mycobacteria nocardia also. I will not read slide by slide to save time. The biggest advantage of MALDI-TOF technology is that it gives you a single choice up to species level for identification. It will not misguide you by giving 4 or 5 multiple choices, and amongst that you have to pick up an organism. Coming to the other advantages of Vitec MS and MALDI-TOF technology. So in Vitec MS, we have 2 separate databases. One is the clinical database and the other one is the RUO database. The benefit of the clinical database is that it is completely brick-walled and fireproof. That means our basic clinical microbiology diagnostic work completely goes on unaffected, whereas the RUO mode gives us the full flexibility and liberty to do all kinds of research-based work, dendrogram analysis, or entering our own spectra, entering our own organism list in case if we want to, that we can do on the RUO. And at the same time, in case if there are some species which are not there in the database, you can work them on the RUO and you can communicate with the bioMérieux team, and they can include this thing in their next or upcoming versions of the database. So this is the benefit. When we talk about the number, when we talk about the MALDI-TOF, it is extremely important that what are the specific things are we talking about. So in MALDI-TOF, there are 3 very important terms that we talk about. First is about the species, then the strains, and the spectra. So many a times when we talk about, there are different modes or there are different ways. Some people talk about the species. Some people say that our database is having these many spectra, or they will have these many strains. So from a clinical microbiology point of view, we should ask 2 things. First is that what are the species which are available, and what is the quality of those species which are available. Secondly, how many spectras were used to make those species, and how many different strain variants were chosen. So I give a very simple example of a classroom. So in a classroom, for example, there are 80 students. So similarly, in a list of MALDI-TOF or in a list of an organism, you will find 80 numbers. But not all the students are brilliant or meritorious, right? About 15 to 20 students will be brilliant. About 60 to 70 students will be average, whereas 4 or 5 students or 5 to 10 students may be below average also. But still they are there in the list. So just looking at the list, whether the list is big or small or whatever, may not be sufficient. So we have to see that what is the list, what is the confidence level behind the list, what is exactly, or is the list a part of the FDA database or not. So bioMérieux, as I said, has got world's largest repository of strains, and we have used on an average up to 12 to 15 strains collected from different geographical location to make the database. Coming to the other part, that means once the identification is done, now what next? Because identification is not the end of the story. So as soon as the identification is there, we should immediately follow the kinetic reporting pattern. So kinetic reporting means that as soon as we get the sample, we inform the clinician that the sample is received. As soon as we get the detection, we should inform that the sample has come positive or negative. As soon as we perform gram staining, we can inform that yes, it's a gram positive or gram negative. Similarly, as soon as the Vitec MS system gives the identification, it has to do 2 things. First, we have to inform the clinician that this is a particular organism which has been identified. So based on that identification, they can choose which antibiotic they can use. So from a broad spectrum antibiotic, they can come down to a narrow spectrum antibiotic when they know the identification. And when they know up to the species level identification, so they can know about the intrinsic resistance. For example, if they know the organism is Stenotrophomonas maltophilia, then even the most expensive antibiotics like meropenem, like colistin, will not work on Stenotrophomonas maltophilia. And very simple, antibiotics like levofloxacin, cotrimoxazole can work. Similarly, if the organism is Brucella, then again the same protocol is used for identification of Brucella, and a completely different course of action can be taken. Similarly, if the organism is Burkholderia pseudomallei, then again a different course of action is required in that case. So as soon as you get to know the right target, so we can choose our weapons. That means what should be the best antibiotic, which antibiotics are to be used, and so on. And by the time we get the antibiotic susceptibility testing, either with the help of Vitek2 or with the help of BioFire, because BioFire can give you the identification and the resistance mechanism detection in less than 45 minutes. So as soon as you get that, then you can go a step ahead of that. So coming to the other parts, that is how exactly the overall thing will benefit. The system, again, as I said, I will keep the things short. So the advantage of the multi-top system is that a single system, regardless of which sample type is to be used, has almost the same protocol. So for all the bacteria, aerobic and anaerobic, the process is same. For mycobacteria and for filamentous fungi, it requires inactivation and extraction. And for Brucella, again, because of the highly pathogenic nature, you have to first inactivate it and then extract and go ahead. So from a technician point of view, because ultimately, no matter how the advanced technology we bring in the lab, ultimately it is the technologists and the technicians who are going to use it. So as Gandhiji has said, that before making any decision, we have to think about the last person who will be impacted because of that. So similarly, whenever we are acquiring any new technology, whether it's a next generation sequencing system or a multi-top system or a full microbiology lab automation or anything, so ultimately we have to see that who is going to use it or on a day-to-day basis. And if the technicians or the engineers, they are not able to use it on a day-to-day basis, then it will be like only a system to showcase in a museum. So we have to ensure that the technicians, the people should be able to use and make the full benefits of the system. So here I close my very short talk. A technology which can drastically reduce the cost of identification. I will not say how much of the cost. Extremely easy workflow, and it can go to the exact identification up to species level, in almost 98.5% of the cases. So I thank the Maharashtra MicroCon. I thank Karyakarte sir and the entire team of for giving us the opportunity to speak. Thank you, sir.
I would like to request Dr. Sunil Bhambre sir to felicitate Mr. Harshul Mishra.